Assalamu alaikum guys. So in today's video we are going to cover the rheumatology chapter from Toronto Notes 2022 edition. So if you open up the rheumatology chapter, you will first see the table of content, which includes the anatomy of joint, pathology, basics of immunology, and differential diagnosis of common presentations. So we'll try to cover the initial part of the rheumatology chapter, that is a revision of your basic anatomy and physiology, I would say. So let me start with what is the basic knowledge that they are covering in the initial part of the chapter. So, in the first part, they want you to differentiate between degenerative and inflammatory joint. So, what do you understand by a degenerative and inflammatory joint? If you cover the first picture that is present in the Toronto Notes Rheumatology chapter, so you'll see there is a joint presentation which has the two bones, and there is this cartilage along with the bones on the joint, and then there is this tendon covering the joint area, and on the upper side there are muscles. Let's assume these are muscles, these are tendons, and this is the cartilage right above the bone, and between all these things are the synovial fluid. So this is basically a gross picture of a joint, basically the gross things that you can see on an anatomy of a joint. So what is the difference between a degenerative and an inflammatory joint? Let's have a conceptual picture of what exactly we need to know about this anatomy. So if you see a bone of a degenerative inflammatory joint, so you will figure out that in a degenerative joint there is a cartilage particle. That means the cartilage is destructed, the cartilage particle, and you would also see there is a loss of joint space. Okay, loss of joint space. That means you will also find that there is a cartilage destruction, and along with that there is an osteophyte, and the formation of osteophyte osteophyte is an overgrown bone. So it's ultimately a bone which definitely would reduce this space in between. So an osteophyte formation will ultimately lead to the loss of joint space, and along with that you see the cartilage destruction or cartilage particles that are present within the joint space. And so this is basically an idea of a degenerative joint. So this is a degenerative joint. And then if we follow up with an inflammatory joint, so this is the bone, this is the another bone, and then along with this you could see that there are cartilage destruction. So you would see that there are cartilage destruction as in the degenerative joint. As in the degenerative joint there is cartilage destruction. Along with that there is this is, let's say this is a tendon, there is effusion, that is the synovial fluid axis. There's effusion, there's this synovitis. Inflammation means itis, so there's this synovitis, and along with that there is a joint space narrowing. So in the previous picture we saw that there was loss of joint space, but in this picture we can see that there is joint space narrowing. Along with that there is also an effusion. Effusion is, as we have discussed here, effusion is an excess of I would say swelling, excess of synovial fluid which leads to swelling. Okay, so this is basically an idea of an inflammatory joint. Inflammatory joint would definitely have inflammatory markers raised, which includes ESR and CRP. And so basically the most commonly used terminologies in rheumatology that you would find are arthritis. Arthritis is arth is the joint and itis is inflammation. So arthritis means inflammation, and along with that there is the erythema, warmth, pain, arthralgias, anything that includes arth. For example, if it's arthritis, arthroscopy, arthralgias, arth means joint related. Okay, and then we will be now revising the difference between a degenerative and an inflammatory joint. So inflammatory, as the name suggests, is is an inflammatory is a condition in which there is an inflammation of the joint and its associated structure. For instance, there is an inflammation of synovium, that is the synovitis. There is effusion, there is joint space narrowing, and there is cartilage destruction. So as you know, you have already studied that the inflammation may be find rubor, that is redness, pain and swelling, and inflammation and erythema. These are all the things that are associated with an inflammatory joint. Likewise, and then if you follow up with, I would say, a degenerative joint, you could see that a degenerative joint would have a cartilage particles, that means the destruction of the cartilage along with loss of joint space and the osteophytes formation. So the only thing that you may remarkably say in a degenerative joint is the osteophytes formation and a loss of joint space. Whereas in the inflammatory joint, you would find joint space narrowing but not the loss of joint space. Along with that, you would also find synovitis and effusion. So I hope this video will give you a brief introduction of the rheumatology that is being covered in the Toronto notes. Now we'll be following up with the basic of immunology, that is also covered right below this part of anatomy of the joint pathology. So let's follow with the basic of immunology. So basics of immunology would cover the four types of immunology reaction that occurs, the mechanism of immune mediated disorders. For instance, there's this type 1 reaction, there's this type 2 reaction, and then type 3 reaction, and type 4 reaction. So type 1 reaction, the immediate hypersensitivity reaction, IgE mediated reaction. So there are other names for type 1 that is known as IgE mediated hypersensitivity reaction mediated, or there could also be like immediate hypersensitivity reaction. So what happens in this reaction is that, as the name suggests, there is an allergen that binds to IgE antibodies on the mast cell. For instance, this is a mast cell which has an IgE antibody. So let's see, this is an IgE antibody, and this is an allergen that binds to, this is an allergen. So the allergen comes up and it binds to IgE antibody, and let's say this is the mast cell on which there is IgE antibody, to which an allergen binds. And when there is this process occurs, there's a release of inflammatory mediators, for example histamine, serotonin, and so on. So this is a brief process of a type 1 hypersensitivity reaction. So if you would ask me examples for this kind of reaction, I would definitely say asthma, an allergic reaction, to a drug. Maybe it can be to a bee sting, or an anaphylaxis reaction that may occur as a reaction to a metal, to something that a person is allergic to while eating. So yeah, so this is the IgE type 1 immediate hypersensitivity reaction, in which the allergen binds to IgE antibody and mast cell release histamine, that occurs in asthma, allergic reactions, and anaphylaxis. Now if we read about the IgE antibody mediated cytotoxic type 2 reaction, which is also known as antibody mediated, or we can also know it is also known as cytotoxic type 2 reaction, in which the IgG or IgM antibody deposits and bind to cell membrane or matrix associated antigen, leading to lysis of the target cell. So there's this cell which is lysed when IgM or IgG antibody deposits and bind to the cell membrane. So there's this IgG antibody or IgM antibody, that is when they deposit onto the cell membrane of the cell. There is the reaction that occurs in the response is the lysis of cell. So it leads to the lysis of the cell. So this is known as cytotoxic. That means cyto means cell, toxic means breakup. So cell breakup, which is due to the IgG and IgM antibodies that deposits onto the cell membrane. So if you read up the names, you'll understand what the mechanism of action of each of the steps of these types of immunology reaction. So in type 1 IgE mediated, there is an allergic reaction where the IgE antibody allergens comes up and bind to the mast cells, and there's a release of the inflammatory mediators, followed by type 2, which is cytotoxic and antibody mediated, in which there is IgG or antibody or IgM antibody that deposits onto the cell membrane and leads to the lysis of the cell. So basically the example for such kind of reactions are, I would say, autoimmune, hemolytic anemia. So in that basically in that disease there is a breakup of red cells, and that is just because of the antibody that binds to the surface of the red cells membrane, and it leads to the breakup of the cell. Along with that, there is an anti-glomerular basement membrane disease, or you may also say Goodpasture syndrome, which also is a reaction of type 2 hypersensitivity reaction. And then there's also Graves disease, and basically these are all the autoimmune disorders that are because of the antibody formation in the blood, which binds to the cell and leads to the lysis or breakup of the cells. Then there is rheumatic fever, and along with that there is idiopathic thrombocytopenic purpura, ITP. So if we follow up with the type 3 hypersensitivity reaction, then we would definitely, let's follow up on type 3 over here on this part of the page. So type 3 is also known as immune complex hypersensitivity reaction. So as the name suggests, so there is antigen antibody complex that deposit in tissues and which activates complement and recruits inflammatory mediators, resulting in tissue injury. So let's see, this is an antigen and antibody complex that deposits within the tissue, and it leads to activation of the complement system. And as it activates the complement system, so there is a recruitment of or there's a release of inflammatory mediators. So as the antigen or antibody complex activates the complement system, there is a release of inflammatory mediators, which results in the tissue injury. So this is another tissue, let's say, so it will definitely injure this. So the example for this kind of reactions are SLE or polyarthritis nodosa, which is a type of vasculitis. And then there is serum sickness and viral hepatitis. And last is the post streptococcal glomerulonephritis. So these are the examples for type 3 immune complex hypersensitivity reaction. Now we'll do the last, we'll cover The last but not the least, that is type 4 hypersensitivity reaction, that is also known as cell mediated or delayed hypersensitivity reaction. So what happened in this type of hypersensitivity reaction is release of cytokines by sensitized T cells and T cells mediated cytotoxicity. For example, there is a release of cytokines by sensitized T cells. This is the T cell that would release, that is once sensitized, would release cytokines and T cell mediated toxicity. Basically, this is the T cell that would release cytokines when is activated, and that would definitely destroy further cells. So the examples for such kind of a hypersensitivity reaction are contact dermatitis, followed by insect venom, mycobacterial proteins, tuberculin skin test, insect venom, contact dermatitis. So these are the examples for hypersensitivity reaction. So if we revise this whole chapter, so we would see in the basics of immunology we have 4 types of immune reaction. Let's say we have type 1 reaction, in which is IgE mediated type 1 reaction, which is also known as immediate hypersensitivity reaction, in which there is a allergen which binds on to the mast cells via IgE antibody, and which leads to the release of histamine. So the examples that are supposed to be covered in this type of hypersensitivity reaction, that is IgE mediated immediate hypersensitivity reactions, are asthma, allergic reactions, and anaphylaxis. And then there is a type 2 antibody mediated reaction, that is type 2 antibody mediated cytotoxic reaction, that is in which there is an IgM antibody that is attached to the cell, and it leads to the lysis of the cell. IgM antibody that attached to the cells and leads to the lysis of the cell. So the reactions that are such reactions happen in autoimmune disease, which are autoimmune hemolytic anemia, Goodpasture syndrome, Graves disease, rheumatic fever, and ITP. So basically these are I would say the autoimmune disease that we usually cover: autoimmune hemolytic anemia, Graves disease. There's an antibody attachment, it leads to the lysis of the cell. Similarly, we'll follow up with the type 3 antibody reaction, that is type 3 hypersensitivity reaction, which is also known as the immune complex. So as the name suggests, there is an immune complex that is made up, that is an antibody and antigen complex that activates the complement, and it leads to a cell destruction. So the type 1 is Ig mediated, which is immediate hypersensitivity reaction. Type 2 is the antibody mediated reaction, which is the cytotoxic. And type 3 is immune complex mediated. So if you learn the second name, I would say that would be more easier to remember exactly what type of reactions are these type of hypersensitivity reaction would have. So if you learn like cell mediated delayed hypersensitivity reaction, the name suggests that it is T cell mediated hypersensitivity reaction in type 4. So let's see, this is the T cell which is attached, and its activation will lead to a release of mediators, which occurs in like let's say contact dermatitis, insect venom, and tuberculin skin test. So yes, this is basics of the immunology that is covered in the Toronto notes. And then if we follow up with the another part of the immunology, that includes the basic functions of different types of cells. For instance, let's just cover it very briefly: innate immune cells, neutrophils, natural killer cells. So neutrophils circulates in the blood, responds to inflammatory stimulus, kill invading organism by phagocytosis, degranulation, and neutrophilic extraction. So likewise, there are multiple different cells mentioned. Along with that, their functions are mentioned, so you need to just briefly get over that part. And now we'll cover the immunogenetics and disease. So the short arm of the chromosome 6 contains gene that encodes HLA molecule, And in human, the HLAs act as an MHC, which presents antigen to the recognized T cells and identify the self by immune system such that they may be matched for in an organ transplantation. So there is an HLA typing that is done for organ transplantation, and then classes of MHC. So there are different classes of MHC, MHC 1, 2, and 3. And along with that, so class 1 has 3 further types that are HLA-A, HLA-B, HLA-C. And then they are all nucleated cells and recognized by the CD cytotoxic T cells. Types MHC type 2 are HLA-DP, HLA-DQ, and DR. Antigen presenting cells, mononuclear B cells recognized by CD4 helper T cells. And then MHC type 3, some components of the complement cascade in plasma, and they function as a chemotaxis, opsonization lysis of the bacterial cell. So HLA associated rheumatic diseases. So there are different type of diseases which specifically are covered in this part of the immunology section. Let us discuss that, because that would really help you to recognize conditions during your license exam. That is the HLA associated rheumatic diseases. So if you cover this part, this will definitely, by the end of this video, you will be definitely be ready for picking up the diagnosis with the cases. For example, if a patient is HLA-B27 positive, so the first thing that would come to your mind is ankylosing spondylitis. That is a seronegative spondyloarthropathy. And then another thing that would come to your mind is the reactive arthritis. That is another seronegative spondyloarthropathy. Another is erosive arthritis. And then there is a psoriatic arthritis. So these are all spondylonegative arthropathies, and the relative risk is 20 times for developing ankylosing and reactive arthritis. So if the person is a HLA-B27 positive, then most likely the person's condition is associated with one of the four conditions. But HLA-B27, as a reminder, is just is a screening test, is not a definitive test for ankylosing spondylitis or any of the conditions. Basically, if a person is positive for HLA-B27, means that a patient most likely has these conditions, but is or may susceptibly have these conditions, but is not definitely going to have these conditions. Now, if we talk about DR4 or DR1, these are the HLA types. So if we talk about DR4 and DR1, then the patient is susceptible to have rheumatoid arthritis, which is also an autoimmune condition, which is 2 to 10 times a patient would have DR4 and DR1 positive, and 93% of the patients with such positivity would have rheumatoid arthritis. And again, we would follow DR3, which is another HLA. And if the patient is positive for DR3, that means the patient may have systemic sclerosis, or the patient may have SLE. So this is another associated HLA that is associated with systemic sclerosis or SLE. So DR3 is associated with the production of anti-Rho SSA and SSB antibodies, which you would definitely find in systemic sclerosis. So DR3 is associated with the anti-Rho, which is also known as SSA, or LA, which is SSB. So SSA usually is found in systemic sclerosis, and along with that, it is also found in neonatal sclerosis, so neonatal lupus. So these are the two conditions that you would find these antibodies. But honestly speaking, the one of the antibody that you would only find in systemic sclerosis is this one. So there's no other associated condition, so this can be a definitive test for systemic sclerosis. So this comes to an end of what we will be covering, what we have covered in the introductory chapter of rheumatology in Toronto notes. So if you go through the previous lecture that I've covered in this entire video, is that we have covered the difference between a degenerative and inflammatory joint. That means a degenerative joint would have loss of joint space and osteophytes formation, whereas an inflammatory joint would have an inflammation along with the narrowing of the joint space and synovitis and cartilage destruction. Following up with that four types of immunology reaction: type 1, type 2, type 3, and type 4. Revising with the type of reactions we would find is IgA mediated in type 1, antibody mediated in type 2, and immune complex in type 3, and T cell mediated in type 4. So it's better to remember their mechanism of action along with few example that these conditions will be associated with. And then we are covering the HLA, three types of HLA types, that is DR that is B27, DR4 and 1, and DR3. So HLA-B27 is associated with ankylosing spondylitis, reactive arthritis, erosive arthritis, and psoriatic arthritis, whereas DR4 and 1 are cover under the rheumatoid arthritis. A patient who positive for DR4 and 1 would possibly may have rheumatoid arthritis, whereas DR3 are positive for systemic sclerosis and SLE. Systemic sclerosis further has anti-Rho, anti-La, SSA, SSB, which is positive for these two conditions. So yes, I hope this video will help you in motivating to read through or cover Toronto notes, which is very difficult for students who are appearing for MCCQE part 1. So my current goal is to cover Toronto notes as much as possible for all of those people who are appearing or planning to appear for MCCQE1. So to support me and to give me motivation, please subscribe to the channel and like the video. Thank you so much.